Hello students, myself Abhilash. In the last class, we already completed about evolution. So in this video, we are going to start with new chapter of second PU biology, that is microbes in human welfare. Microbes in human So, if you see the chapter, it has two words, that is microbes and another one is human welfare. What are microbes? Microbes are also known as microorganisms. ಹಾಗಾದರೆ microbes ಅಥವಾ microorganisms ಅಂದ್ರೆ microbes or microorganisms, those organisms which are cannot visible through our naked eyes. They are known as microbes or microorganisms. The organisms which we cannot visible through our naked eyes. Naked eyes ಅಂದ್ರೆ ಯಾವ ವಸ್ತುಗಳು ಅಥವಾ ಯಾವ ಜೀವಿಗಳು ನಮ್ಮ ಬರಿಯ ಗಣ್ಣಿಗೆ ಕಾಣುವುದಿಲ್ಲವೋ, ಗೊತ್ತಾಗ್ತಿದೆಯಾ? which we cannot visible through our naked eyes, they are known as microbes or microorganisms. ಈಗ ಅವನು ನಾವು ಬರಿಗಣ್ಣಲ್ಲಿ ನೋಡೋದಕ್ಕೆ ಆಗೋದಿಲ್ಲ, but we have to study their nature and characters. To study or to visible microbes, we have a special instrument known as microscope. This microscope helps to visualize the microorganisms. Now already microscope na use ಮಾಡಿದೀವಿ. Yeah, it has lenses through which we undergo or we going to study, visualize those smaller creatures. Okay, and in this chapter we are going to study about the beneficiary or good things of microbes in our day-to-day activities. ಹಾಗಾದ್ರೆ ಮೈಕ್ರೋಬ್ಸ್ ಇಂದ ಕೆಲವೊಂದು ಮೈಕ್ರೋಬ್ಸ್ ಇಂದ ನಮಗೆ ಉಪಯೋಗಗಳಿವೆ. ಆ ಉಪಯೋಗ ಕೊಡುವ ಮೈಕ್ರೋಬ್ಸ್ ಯಾವ್ಯಾವು, ಅದರ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಏನು, ಯಾವ ರೀತಿ ನಾವು ಅದರಿಂದ ಉಪಯೋಗ ಪಡ್ಕೊಳ್ತಾ ಇದ್ದೀವಿ ಅನ್ನೋದನ್ನ ಈ ಚಾಪ್ಟರ್ ಡೀಟೇಲ್ಡ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡ್ತೀವಿ. ಮೈಕ್ರೋಬ್ಸ್ ಹ್ಯಾವಿಂಗ್ ಗುಡ್ ಸೈಡ್ ಆಲ್ಸೋ, ಬ್ಯಾಡ್ ಸೈಡ್ ಆಲ್ಸೋ. In this chapter, we are going to focus only on good side of microbes. And these microbes, they are already smaller in size, and we use microscope to visualize those microbes. Next, microbes plays a special compartment, or microbes plays a major role in biological systems. Microorganisms plays a major role in biological system. Biological system which includes plants and animals. Okay, natural system ಅಲ್ಲಿ plants ಮತ್ತು animals ಕೂಡ ಇದೆ. ಅದರ ಜೊತೆ ಮೈಕ್ರೋಬ್ಸ್ ಕೂಡ ಬಹಳ ಇಂಪಾರ್ಟೆಂಟ್. ಹೇಗೆ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂದ್ರೆ ದೀಸ್ ಮೈಕ್ರೋಬ್ಸ್ ಆರ್ ಮೋರ್ ಅಂಡ್ ಮೋರ್ ಇನ್ ನಂಬರ್ ಕಂಪೇರ್ಡ್ ಟು ಪ್ಲಾಂಟ್ಸ್ ಅಂಡ್ ಅನಿಮಲ್ಸ್. ಈ ಮೈಕ್ರೋಬ್ಸ್ ನ ಸಂಖ್ಯೆ ಇರುತ್ತೆ, ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಯಾರಿಗೆ ಕಂಪೇರ್ ಮಾಡಿದ್ರೆ ಪ್ಲಾಂಟ್ಸ್ ಮತ್ತೆ ಅನಿಮಲ್ಸ್ ಕಂಪೇರ್ ಮಾಡಿದ್ರೆ, ಬಟ್ ಯು ಕೆನಾಟ್ ಸಿ ಡೈರೆಕ್ಟ್ಲಿ. ಅದು ನಾವು ನೇರವಾಗಿ ನೋಡೋದಕ್ಕೆ ಆಗ್ತಾ ಇಲ್ಲ, ಅಷ್ಟೇ ಹೊರಟ್ರೆ ಅದರ ಪಾಪ್ಯುಲೇಷನ್, ಅದರ ಸಂಖ್ಯೆ ಏನಿದೆ, they are more in number. More in number means These undergo reproduction very fast. ಈ ಮೈಕ್ರೋಬ್ಸ್ ಅಲ್ಲಿ ರಿಪ್ರೊಡಕ್ಷನ್ ರೇಟ್ ಏನಾಗಿರುತ್ತೆ, ತುಂಬಾ ಫಾಸ್ಟ್ ಇರುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಇಫ್ ವಿ ಟೇಕ್ bacteria. Bacteria is also microbe, belongs to kingdom Monera. This bacteria, the type of bacteria we are going to take an example of is E. coli, that is Escherichia coli. This bacteria undergo several times reproduction within 1 hour. 1 hour ಏನಾಗುತ್ತೆ, Escherichia coli ಇಷ್ಟೊಂದು ಸಲ, ತುಂಬಾ ಸಲ ಏನಾಗುತ್ತೆ, ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ. ರಿಪ್ರೊಡಕ್ಷನ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, ದ ರೇಟ್ ಆಫ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಈಸ್ ಮೋರ್. Okay, hence the offspring is produced more means the population size also increases. Okay, this is about the role of microbes in biological system. And very important thing is the microbes are cosmopolitan in distribution. Microbes are cosmopolitan in distribution. Cosmopolitan. Cosmopolitan means present everywhere. ಎಲ್ಲಾ ಕಡೆನೂ ಇರುತ್ತೆ. ಉದಾಹರಣೆಗೆ ಸರ್ವಾಂತರ್ಯಾಮಿ ಅಂತ ಏನು ಹೇಳ್ತೀವೋ ಅದೇ ರೀತಿ cosmopolitan. Why? Because the microbes are found in sand, they are found in water, they are found in air also, they are found in rocks also. Okay, like this, the microbes found in snow also. Okay, that's why we say that microbes are cosmopolitan in distribution. These microbes are found in, right here, some microbes are found in deep sea, thermal vents. Thermal vents ಅಂದ್ರೆ ಯಾವ ಪ್ರದೇಶದಲ್ಲಿ ಟೆಂಪರೇಚರ್ 100 ಡಿಗ್ರಿಗಿಂತ ಜಾಸ್ತಿ ಇರುತ್ತೋ ಗೊತ್ತಾಗ್ತಿದೆಯಾ, ಅದನ್ನ ಥರ್ಮಲ್ ವೆಂಟ್ಸ್ ಅಂತ ಕರೀತೀವಿ. ಸಮುದ್ರದ ಆಳದಲ್ಲಿ ಕೆಲವೊಂದು ರೀಜನ್ ಅಲ್ಲಿ ಟೆಂಪರೇಚರ್ 100ಕ್ಕಿಂತ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಅಲ್ಲೂ ಕೂಡ ಈ ಮೈಕ್ರೋಬ್ಸ್ ಬದುಕಿರುತ್ತೆ. And some microbes, they are live in snow, ಹಿಮ ಬೀಳುವ ಪ್ರದೇಶದಲ್ಲಿ, where we can see that the temperature is -40 degree or -30 degree Celsius. In that places also we observe microbes live their life. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? ಹಾಗಾದ್ರೆ ಇಷ್ಟು ಟೆಂಪರೇಚರ್ ಕಡಿಮೆ ಇರುವ ಪ್ರದೇಶದಲ್ಲಿ ಅಥವಾ ಇಷ್ಟು ಟೆಂಪರೇಚರ್ ಜಾಸ್ತಿ ಇರುವ ಪ್ರದೇಶದಲ್ಲಿ ಹೇಗೆ ಮೈಕ್ರೋಬ್ಸ್ ಬದುಕುತ್ತೆ ಅಂದ್ರೆ, ದೇ ಹ್ಯಾವ್ ಸಮ್ ಅಡಾಪ್ಟಿವ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? ಒಂದೊಂದು ರೀತಿಯ ಮೈಕ್ರೋಬ್ಸ್ ಅಲ್ಲಿ ಒಂದೊಂದು ಅಡಾಪ್ಟಿವ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಇರುತ್ತೆ. ಥ್ರೂ ದಟ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ದೇ ಆರ್ ಸರ್ವೈವ್ ಇನ್ ಎನಿ ಏರಿಯಾಸ್. And we gave one more name to microbes. Some microbes, they are known as thermoacidophils. Thermoacidophils. This is a single word, but we can split into 3 words: thermo, acido, and phils. First, phili means love or attraction. Some microbes live in high temperature, okay, whereas some microbes are live in high acid concentration also. So those are known as thermoacidophils. Okay, this is a small introduction of microbes. Then after that we are move on with the next concept. Next, we are move on with Diversity of microbes. There are various diversities are seen in microbes. For example, if we take bacteria, they are also microbes. Bacteria belongs to kingdom Monera, first kingdom, and this bacteria is of two types. One is Archibacteria and another one is Eubacteria. And next, if we see fungi, they are belongs to special kingdom also known as kingdom fungi, or we can also known as kingdom mycotas. Then, if we see the diversity of microbes, the next we will discuss is virus. The next we are going to study is viruses. Study of virus is known as virology, and these viruses are smaller than bacteria. Nodi bacteria plural, bacteria singular. Okay, these viruses are smaller than bacteria. Bacteria kina chikkadirutte. If we take a typical virus structure by taking T4 bacteriophage, which we have already learned in our first PU. This is the structure of T4 bacteriophage. I write roughly. This T4 bacteriophage, if we label them, this is T4 bacteriophage. It is not a bacteria, it is a virus actually. This is known as the head, this is known as collar, and this is the tail, and these are known as tail fibers. Okay, and these smaller structures are said to be the plates. This is a rough structure I write to show a bacteriophage, that is a virus. If we see the viruses have two major properties. If we see the properties of viruses, the first thing is the genetic material that found in viruses is either DNA nor RNA. The genetic material in viruses is either DNA nor RNA, but not two. ಎರಡರಲ್ಲಿ ಒಂದಿರುತ್ತೆ, ಎರಡು ಇರೋದಕ್ಕೆ ಪಾಸಿಬಿಲಿಟೀಸ್ ಇಲ್ಲ. And second thing is, these viruses which consist nucleic acid, either the nucleic acid in viruses is either single stranded or double stranded. Single stranded allu irbodu, double stranded allu irbodu. Nammali DNA is a genetic material which is double stranded. Other riti it depend upon viruses whether they have contained DNA or RNA as genetic material. whether the nucleic acid is single or double stranded. Okay. After the viruses, we will look off, and one more thing is, if we see the head portion and it has the genetic material, and remember here, thought this is a genetic material, and this genetic material is covered by a protein coat known as capsid. In case of viruses, the genetic material is covered with a protein coat known as capsid. This is genetic material and this is capsid. The capsid has small compartment. You can see these small compartments. These small compartments are known as capsomeres. These smaller compartments, we call them as capsomeres. Okay. Next, after viruses, we move on with viroids. Here on the board. Remember, we completed bacteria and we move on with viruses. Bacteria is bigger and viruses are smaller. Next, we will discuss about viroids. Viroids are smaller than viruses. ಈ ವೈರಾಯ್ಡ್ಸ್ ಏನಿದೆ ವೈರಸ್ ಗಿನ ಚಿಕ್ಕದಿರುತ್ತೆ. ಹಾಗಾದ್ರೆ ವೈರಾಯ್ಡ್ಸ್ ಅಂದ್ರೆ ಏನು? The viruses which devoid of capsid or protein coat. ನಾನು ಹೇಳಿದೆ ವೈರಸಸ್ ಅಲ್ಲಿ ಪ್ರೋಟೀನ್ ಕೋಟ್ ದಟ್ ಇಸ್ ಕ್ಯಾಪ್ಸಿಡ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ಅಂತ. ಬಟ್ ಈ ವೈರಾಯ್ಡ್ಸ್ ಅಲ್ಲಿ ಆ ಪ್ರೋಟೀನ್ ಕೋಟ್ ಕ್ಯಾಪ್ಸಿಡ್ ಏನಿದೆ ಅದು ಆಬ್ಸೆಂಟ್ ಆಗಿದೆ. ಇನ್ ವೈರಸಸ್ protein coat that is capsid is absent. So in viroids the protein coat that is capsid is absent. ಅದಕ್ಕೆ ಹೇಳ್ತಾರೆ viroids means the viruses that naked with protein coat. ಯಾವ substances ಅಲ್ಲಿ protein coat naked ಇದೆಯೋ they are known as viroids ಅಂತ ಹೇಳ್ತಾರೆ. And third one is prions. We already know about prions. Prions are infectious proteinaceous agents. Prions are infectious proteinaceous agent. Okay, this is about a general things before we start with the chapter. Ella early information. And one more thing, we told these are smaller, we cannot visible through our naked eyes. But to study about these in detail, these microbes in detail, in laboratory we are going to culture these microbes. Now lab ಅಲ್ಲಿ ಏನು ಮಾಡ್ತೀವಿ, ಈ microbes ನ ಕಲ್ಚರ್ ಮಾಡ್ತೀವಿ by using culture media. ಕಲ್ಚರ್ ಮೀಡಿಯಾ ಪ್ರಿಪೇರ್ ತೆಗೆದುಕೊಂಡು ಏನು ಮಾಡ್ತೀವಿ, ಲ್ಯಾಬ್ ಅಲ್ಲಿ microbes ನ ಕಲ್ಚರ್ ಮಾಡ್ತೀವಿ. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? And to culture these microbes, the culture media mainly consists of culture media ದಲ್ಲಿ ಏನೇನು ಬಳಸ್ತೀವಿ ಅಂದ್ರೆ ಅಗರೋಸ್ ಬಳಸ್ತೀವಿ, ಆಮೇಲೆ ಪೊಟ್ಯಾಟೊ ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇದೇ ಒಂದು ಪೆಟ್ರಿ ಪ್ಲೇಟ್ ಅಂತ ಅಂದ್ಕೊಂಡ್ರೆ ನಾವಿಲ್ಲಿ ಕಲ್ಚರ್ ಮೀಡಿಯಾನ ಹಾಕ್ತೀವಿ. ಸ್ವಲ್ಪ ದಿನ ಬಿಟ್ಟ ಮೇಲೆ ದೀಸ್ ಮೈಕ್ರೋಬ್ಸ್ ದೇ ಆರ್ ಲಿವಿಂಗ್ ಕಾಲೋನೀಸ್. ಈ ರೀತಿ ಏನಾಗಿರುತ್ತೆ, ಅವು ಡೆವಲಪ್ಮೆಂಟ್ ಆಗಿ ಏನಾಗಿರುತ್ತೆ, ಕಾಲೋನಿ ಫಾರ್ಮೇಶನ್ ಮಾಡಿರುತ್ತೆ. ದಿಸ್ ವಿ ಕ್ಯಾನ್ ವಿಸಿಬಲೈಸ್. ಇದನ್ನು ನಾವು ಆರಾಮಾಗಿ ನೋಡಬಹುದು. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? This is how we culture microbes in laboratory, that is in artificial medium. Okay, and in this chapter I already told we are going to study about only the beneficiary activities or things of microbes that is related to humans. So let's start the chapter with beneficiary activities of microbes to humans. The first thing we are going to study about use of microbes in household purposes. Next, we are going to study the use of microbes in household applications. That is, microbes in household applications. In that, various microbes take part in our day-to-day life activities. Let's discuss a few microbes that are involved in our day-to-day life activities. The first we are going to study about the microbe lactobacillus. As the name indicates, lacto means lactose. Lactose is a milk sugar, it is a carbohydrate. Bacillus means a bacteria which is rod-shaped in structure. Bacillus is a rod-shaped bacteria. Lactobacillus is also called LAB. LAB stands for lactic acid bacteria. LAB stands for lactic acid bacteria. What is the use of this lactobacillus or LAB? This lactobacillus or lactic acid bacteria helps in the conversion of milk into curd. This helps in the conversion of milk into curd. ಹಾಲನ್ನ ಮೊಸರಾಗಿ ಕನ್ವರ್ಟ್ ಮಾಡೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡೋದು ಯಾವುದು ಅಂದ್ರೆ ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಆಲ್ರೆಡಿ ಮುಂಚೆನೇ ಹೇಳಿದೆ, lactobacillus is also known as lactic acid bacteria. Lactose is a milk sugar. This lactose is a disaccharide. This is a disaccharide obtained by the combination of glucose and galactose. Glucose and galactose. These two are the monomers, okay, monosaccharides which combine to form a disaccharide, that is lactose. How the lactose helps in conversion of milk into curd? ಹೇಗೆ milk na curd ಆಗಕ್ಕೆ help ಮಾಡುತ್ತೆ ಅಂದ್ರೆ, this lactic acid bacteria contains some acids. ಇದರಲ್ಲಿ acids ಇರುತ್ತೆ. These acids coagulate. These acids these lactic acid bacteria synthesis some acids or secrete some acids. These acids coagulate partially digested milk proteins. These coagulates partially digested milk proteins. So these acids secreted by LAB, they will coagulate clump. They will coagulate partially digested milk proteins. After coagulation ಆದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ? After coagulation, the partially digested milk proteins or milk converted into curd. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? ಸೊ ಹೇಗೆ ಮಿಲ್ಕ್ ಕರ್ಡಾಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ದಿಸ್ ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕನ್ಸಿಸ್ಟ್ಸ್ or secretes some acids. That acids coagulates the partially digested milk proteins. After coagulation ಆದ ನಂತರ after coagulation that partially digested milk converts into curd. ಹಾಲು ಏನಾಗುತ್ತೆ ಮೊಸರಾಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಅಂಡ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್, ಹೇಗೆ ನಾವು ಹಾಲನ್ನು ಮೊಸರಾಗಿ ಕನ್ವರ್ಟ್ ಮಾಡ್ತೀವಿ ಅಂದರೆ ಫಸ್ಟ್ ಏನು ಮಾಡ್ತೀವಿ, ಹಾಲಿಗೆ ನಾವು ಸ್ವಲ್ಪ ಮೊಸರು ಹಾಕ್ತೀವಿ ಅಥವಾ ಸ್ವಲ್ಪ ಮಜ್ಜಿಗೆ ಹಾಕ್ತೀವಿ. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? ಸೊ ಅದನ್ನು ಹೆಪ್ಪಾಕೋದು ಅಂತ ಹೇಳ್ತಾರೆ. ಟು ಪ್ರಿಪೇರ್ ದ ಕರ್ಡ್, ಟು ಮಿಲ್ಕ್ ವಿ ಆ್ಯಡ್ a small quantity of curd or buttermilk. That curd consists of LAB. ಹೌದಾ? ಅದರಲ್ಲಿ ಸ್ವಲ್ಪ ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದ್ದೇ ಇರುತ್ತೆ. ಸೊ that curd that add to milk, okay, that small amount of curd to add milk, that is become inoculum ಅಂತ ಕರೀತಾರೆ. Inoculum or starter ಅಂತ ಕರೀತೀವಿ. ಹೇಗೆ ಹಾಲನ್ನು ಮೊಸರು ಕನ್ವರ್ಟ್ ಮಾಡಬೇಕಾದ್ರೆ ಸ್ವಲ್ಪ ಸ್ಮಾಲ್ ಕ್ವಾಂಟಿಟಿ ಆಫ್ ಮೊಸರು ಆ್ಯಡ್ ಮಾಡ್ತೀವಿ ಅಥವಾ ಮಜ್ಜಿಗೆ ಆ್ಯಡ್ ಮಾಡ್ತೀವಲ್ವ, ದಟ್ ಈಸ್ ಬಿಕಮ್ ಇನಾಕ್ಯುಲಮ್ ಆರ್ ಸ್ಟಾರ್ಟರ್. ಅದನ್ನು ಆ್ಯಡ್ ಮಾಡ್ತೀವಿ, ನೈಟ್ ಒಂದು ಆ್ಯಡ್ ಮಾಡಿ ಬಿಟ್ಟಾಗ ಏನಾಗುತ್ತೆ, ಬೆಳಗ್ಗೆ ಟೈಮಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಮೊಸರಾಗಿ ಕನ್ವರ್ಟ್ ಆಗಿರುತ್ತೆ. ಗೊತ್ತಾಗ್ತದೆ? ದಿಸ್ ಇಸ್ ದ ಒನ್ ಫಂಕ್ಷನ್ ಆಫ್ ದಿಸ್ LAB, that is lactic acid bacteria or lactobacillus. The second function of LAB is this LAB, that is lactobacillus or lactic acid bacteria, helps in increasing vitamin B12 concentration in curd. Increases vitamin B12 concentration in curd. This is the second function of LAB. And third function of LAB is this lactic acid bacteria checks the disease causing microbes activity in the curd. So ಏನಾಗುತ್ತೆ, this LAB checks the activity of disease causing microbes in ಮೊಸರಲ್ಲಿ. ಈ LAB ಬಿಟ್ಟು ಬೇರೆ ಕೂಡ ಮೈಕ್ರೋಬ್ಸ್ ಇರುತ್ತೆ, ಕೆಲವೊಂದು ಡಿಸೀಸ್ ಕಾಸಿಂಗ್ ಮೈಕ್ರೋಬ್ಸ್ ಇರುತ್ತೆ, ಅದರ ಆಕ್ಟಿವಿಟಿನ ಚೆಕ್ ಮಾಡುತ್ತೆ. ಯಾವುದು ಅಂದರೆ LAB. Okay, this is about the lactic acid bacteria that is usually in household applications. Next, we move on about the dough, that is dough. This dough is mainly obtained by fermentation process. ನಾವು ಯಾವಾಗ ಹೇಳ್ತೀವಿ fermentation process ಇಂದ obtain ಮಾಡ್ತೀವಿ. The dough is obtained by the fermentation process used in the preparation of batter for idli and dosa. Okay, ನಾವು ಮನೇಲಿ ಇಡ್ಲಿ ಅಥವಾ ದೋಸೆ ಪ್ರಿಪೇರ್ ಮಾಡಬೇಕಾದ್ರೆ ಅಷ್ಟೊಂದು ಹಿಟ್ಟನ್ನು ಪ್ರಿಪೇರ್ ಮಾಡ್ತೀವಿ. ಅದಾಗ್ತಿದೆಯಾ, ಅದನ್ನು ರಾತ್ರಿ ಹೊತ್ತು ಹಾಗೆ ಬಿಡ್ತೀವಿ. ಅದು ಯಾವ ಪ್ರೋಸೆಸ್ ಆಗುತ್ತೆ ಅಂದರೆ ಇಲ್ಲಿ ಫರ್ಮೆಂಟೇಷನ್ ಪ್ರೋಸೆಸ್ ಆಗುತ್ತೆ. ಅಂಡ್ ಇಫ್ ಯು ಅಬ್ಸರ್ವ್ some dough, the dough is puffy in nature. ಉಬ್ಬಿರುತ್ತೆ, ಅಬ್ಸರ್ವ್ ಮಾಡಿರ್ಬೋದು. This puffy nature of dough is due to the release of a gas known as carbon dioxide. That's some dough have puffy nature. That puffy nature of dough is due to the gas known as CO2, carbon dioxide. Next, if we observe the bread, now bread and observe the bread is obtained by the fermentation of a yeast. Bread na yav riti obtain ಮಾಡ್ತೀವಿ, yav riti ಉಟ್ಕೊಂತೀವಿ ಅಂದ್ರೆ it is obtained or prepared by the fermentation of baker's yeast. Next application is bread. Bread, it is obtained by the fermentation of baker's yeast. This bread is obtained by the fermentation of baker's yeast. Yeast is a unicellular structure. The yeast is belong to kingdom fungi. All fungi are multicellular except this yeast. And the baker's yeast scientific name is Saccharomyces. Saccharomyces cerevisiae. Saccharomyces cerevisiae. This is a scientific name of this yeast. Okay. Next, after next, then toddy. It is a traditional drink. It is a traditional drink. Toddy is a traditional drink that found in the regions of South India. South India, ನಮ್ಮ ಭಾಗದಲ್ಲಿ ಒಂದು drink ಇದೆ. The drink is known as toddy. This toddy is obtained by fermentation of sap in palm tree. The palm tree ಅಲ್ಲಿ sap ಇರುತ್ತೆ. Sap is a nutritive part. ಈ ಪಾಮ್ ಟ್ರೀಯ ನ್ಯೂಟ್ರಿಟಿವ್ ಪಾರ್ಟ್ ಇಂದ ಒಂದು ಲಿಕ್ವಿಡ್ ಅಥವಾ ಒಂದು ಡ್ರಿಂಕ್ ನ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡ್ತೀವಿ ಬೈ ಫರ್ಮೆಂಟೇಷನ್. ದಟ್ ಡ್ರಿಂಕ್ ಈಸ್ ನೌನ್ ಆಸ್ toddy. Okay, toddy is a traditional drink that found in the region of southern India or South India, which is obtained by fermentation of sap in palm tree. Okay, next, after toddy we move about cheese. Actually, cheese are various types or various forms. It depends upon color, flavor. Okay, and cheese are various types in nature. And there is one special type of cheese known as Swiss cheese. This Swiss cheese is obtained by a bacteria. Swiss cheese is obtained by a bacteria known as Propionibacterium shermani. This Swiss cheese is obtained by a bacteria. The scientific name of that bacteria is Propionibacterium shermani. It is a scientific name of that bacteria. Okay, I write this bacteria name bigger. This cheese consists of bacteria known as Propionibacterium Shermani. This is the scientific name of bacterium, so we write in two words. The first word is genus, it should be starts with capital letter, and the second word is species, it should be starts with small letter. And we have one more type of cheese known as Roquefort cheese. Roquefort cheese. This cheese is obtained by fermentation, but the fermentation of fungi. This cheese na bacteria obtained ಮಾಡ್ತೀವಿ fermentation, but this Roquefort cheese is obtained by the fermentation of fungi. Okay, the fermentation of fungi. And one more, I already told about in case of toddy, it is obtained by fermentation of sap in palm trees. We also undergo fermentation in case of fish, in case of soya also. Okay, this is about the today's class. Okay students, so we start with introduction to this chapter, that is what are microbes, diversity of microbes, size, types and placement of microbes in various levels. After that we move on with some types of microbes, that includes bacteria, viruses, viroids, prions. Then we move on with our chapter, that is use of microbes in household applications or purposes. This question is may ask for 3 marks sometimes, or may ask for 5 marks in our board exams. Okay, so the question may be, mention the use of microbes in household applications. If they ask for 3 or 5 marks, we should write 6 points, 6 to 7 points are there. The first point is the lactic acid bacteria or lactobacillus that helps in conversion of milk into curd. Okay, and second point, LAB helps in increases the concentration of vitamin B12 in curd. The third point is LAB also checks the activity of disease causing microbes in curd. The fourth point is some dough are prepared by fermentation process. It is used in the preparation of idli and dosa. Next we use the baker's yeast, that is Saccharomyces cerevisiae, which is used in the preparation of bread in bakery. Next we also mention toddy is a traditional drink which is found in South India, which is obtained by the fermentation of sap in palm trees. Swiss cheese is obtained by the bacteria, that is Propionibacterium shermani. And Roquefort cheese, it is also obtained by fermentation of fungi. Okay, this completes the today's class. Thank you.